is Theodore, and I'll be doing my 3-minute paper presentation on this article titled Biophysical Properties of the Clinical Stage Antibody Landscape. So the authors of this paper aim to analyze whether a current repertoire of approved therapeutic antibodies, as well as antibodies in advanced clinical trial stages, at least phase 2 or above, actually possess the necessary developability, drug likeness, or viability, simply stated. And the traits of developability, among others, as defined by the authors, are high-level antibody expression, high solubility, and covalent integrity. And for small molecules, a metric already exists that quantifies developability. It's known as Lipinski's Rule of 5. And the authors aim to help facilitate the development of an analogous metric for antibodies. And to accomplish this, the authors expressed each of 137 antibodies of interest, as mentioned, that were in advanced clinical trial stages or already approved for therapeutic use. They expressed them as a human IgG1 isotype. They didn't express the exact antibodies, as that would have been too difficult, so they relied on variable domain comparisons to create highly representative antibodies, if I understood correctly. They then subjected those antibodies to a variety of assays, including ACSINS, PSR, polyspecificity reagent, and BVP, baculovirus particle, among others. The acronyms for all of the assays are listed at the top of each of the histograms in Figure 1. And from those assays, they discovered that a few of the analyzed antibodies, although fortunately not the majority, and quite a small number, exhibited lackluster results for developability. For instance, some antibodies, even in stage 3 clinical trials, yielded high results on the PSR assay, which is undesirable since you don't want a polyspecific and very promiscuous antibody. You want it to have high specificity to one antigen. Fortunately, they also discovered that overall the undesirable measurement percentage for all of the assays decreased significantly the more advanced the clinical trial stage was. For instance, approved drugs had the lowest number of antibodies of concern, or warning flags. So the clinical trials were doing their job. The warning flags were established so that the antibodies that scored worse than 90% of the approved antibodies were given a red flag marker. And overall, the authors concluded that they weren't too concerned about their results, since the decrease in poor scores as the clinical trial stage progressed was quite significant, but they concluded that for each set of antibodies in testing, at least about a dozen interconnected developability assays should be conducted prior to the clinical trial stages, and certain cutoffs should be established from those developability assays that identify antibodies that warrant further analysis and perhaps engineering.